Okay, hello everyone. Really want to thank the organizers for inviting me to give this talk. I'm a little bit under the weather, so my apologies. And also I'm going to go through some highlights because there's a lot to cover and obviously just 15 minutes. So if you would like to ask additional questions, if we don't have time, I'm happy to do that after the session. Okay, so my conversation today will be dedicated to cilta-cel, which is a structurally differentiated CAR T cell therapy targeted against BCMA for multiple myeloma patients. And I do want to draw your attention specifically to the fact that this CAR T is targeting two BCMA binding domains that will help to confer high avidity against the human BCMA. I do also want to mention that this CAR T was approved in 2022 for heavily pretreated relapse refractory multiple myeloma patients as based on the CARTITUDE-1 study that I'm going to show here, but it is now also approved for early lines of therapy, which is the CARTITUDE-4 study, which I will cover towards the latter end of my talk. So what I would like to highlight here is mostly the fact that these are heavily pretreated patients that were treated, that were treated previously with 3 or more lines of therapy before they entered the trial. And this is pretty much a standard protocol. And I also want to mention the fact that there are some patients that are part of the CARTINIO study, which is a 15-year follow-up study. Remarkable data as published previously from the CARTITUDE-1 study, and essentially cilta-cel has emerged as a transformative single infusion therapy for these heavily pretreated relapse refractory multiple myeloma patients with almost 100% response rate, 82.5% stringent CR, so very deep responses and also very durable with a median PFS of 33 to 34 months so far. Taking a deep dive into the biomarker data, as this talk is dedicated to that. So we first and foremost looked at the CAR T-cell product, and what we've noticed is that the CAR T-cell product showed preferential expression or expansion of CD8 CAR T positive T cells post-infusion as compared to pre-infusion, and the dominant phenotype is the central memory phenotype of both the CD4s and the CD8 post-infusion.